Hi everyone, my name is Tsotakos and I am an assistant professor of biology at Penn State, Harrisburg. I want to give you a brief description of one of the projects that my lab is currently working on. The focus of the project is diabetic kidney disease. One of the hallmarks of diabetes is proteinuria, which is the loss of the proteins through the urine. You may have heard that the kidneys are responsible for filtering the blood. But how do they do this exactly? They use a specialized type of cell which is called podocyte. Podocyte is a term of Greek origin that means cell that has feet. These cells look like they have feet. These feet branch out of the cell like the fingers in our hands. And these feet from neighboring cells interdigitate to form a filtration device called the slit diaphragm. Think about when you try to make a sandcastle on the beach with your hands. You can cross your fingers to carry the wet sand and the excess water will flow through your fingers, but the sand will stay in your palms. This is what the podocytes in the kidneys do. They filter the blood, keeping the useful materials like the red blood cells and proteins in the bloodstream while getting rid of other components like salts and excess glucose, drug metabolites, etc. In the lab that I used to work in when I was a graduate student, we established a cell culture model that mimics the behavior of these cells under diabetic conditions. In diabetes, the prolonged high levels of glucose in the blood are toxic to the podocytes, and the podocytes eventually die, leading to proteinuria, or, like I said before, loss of proteins to the urine, and the proteins are useful molecules. Now, the loss of podocytes in diabetes does not happen early on. It actually takes many years, sometimes decades, for the podocytes to exhibit loss of function. But this does not mean that during this time the cells are completely fine. In fact, we showed that there are structural changes happening to these cells. And these changes mean that the slit diaphragm gets lost. Now, these changes happen really slow and are very gradual. But once they have happened, just returning the cells to normal glucose levels is not enough by itself for the cells to recover. And you need to remember that in a cell culture system, the cells keep dividing. And even when we change the glucose back to normal levels, they don't reset, so to say. They cannot form new slit diaphragms. So the current focus of my research is to find out ways that we can stop these changes from happening before they happen. This means that we have to find molecules that change early on, when the cells are first exposed to high glucose, and these molecules should be responsible for the irreversible changes that we observe following the long-term exposure to high glucose. We are focusing on a group of molecules that are called long non-coding RNAs. Up until a decade ago, these molecules were not very well known, but more recently, researchers have started realizing their potential. We're looking into the levels of few of these molecules in our cell system, and we monitor these levels after exposing the cells to high glucose. We have found that some of these molecules change very soon after high glucose has been introduced to the system, and definitely before we can see any changes in the function of these cells, any sort of loss of the slit diaphragm. Now we're trying to understand the importance of these events and the association of these events with the loss of the cells in diabetes, to try and find out a way to prevent these changes from happening. We hope that even a delay in the appearance of the symptoms can improve the life quality of the patients, an event which would also save millions of dollars from the insurance system. I hope that you found my research interests as fascinating as I do, and you are more than welcome to get in touch with me to discuss these in greater detail. Thanks for listening.